Hi everyone, welcome to NEET Biology Channel. To study pollen germination, a student should use A. Dry slide, B. Ethanol slide, C. Sugar solution on slide, D. Saline water. Correct answer is C. Sugar solution on slide. Explanation: pollen germination requires a nutrient-rich and hydrating environment, which is provided by a 10% sugar solution. The sugar acts as an energy source and maintains osmotic balance, stimulating pollen tube growth. A dry slide lacks the necessary moisture and nutrients, so no germination would occur. Ethanol, being a dehydrating agent, would kill the pollen grains. Saline water does not provide sufficient energy or the right osmotic conditions for proper germination. In emasculation technique, what is removed?
A. Stigma, B. Ovary, C. Anthers, D. Sepals. Correct answer is C. Anthers.
Explanation: emasculation involves removing the anthers from a bisexual flower to prevent self-pollination and ensure cross-pollination using pollen from a desired male parent. The stigma is essential for receiving pollen and must be retained. The ovary is the site of fertilization and seed development, so it is not removed. Sepals serve as protective structures and do not influence fertilization, thus their removal is unnecessary in emasculation.
Bagging is done to A. Provide warmth to flower, B. Stop pollinator visits, C. Enhance growth, D. Add moisture. Correct answer is B. Stop pollinator visits.
Explanation: bagging is a technique used in artificial hybridization to prevent contamination by unwanted pollen, primarily carried by insects or wind. This ensures that only the selected pollen fertilize the stigma. It is not intended to provide warmth, which could disrupt normal development. It does not enhance growth nor contribute moisture. Its sole function is to isolate the flower from external pollen sources. When is rebagging done during artificial hybridization? A. Before emasculation, B. After pollination, C. Before stigma is receptive, D. After fertilization. Correct answer is B. After pollination. Explanation: rebagging is done immediately after the desired pollen has been applied to the stigma to prevent any unwanted pollen from interfering. Doing it before emasculation serves no purpose, as the flower is still at risk of self-pollination. Rebagging before the stigma is receptive means the pollination itself may fail. After fertilization, the role of isolation is largely complete, making rebagging redundant at that point. Which structure helps guide the pollen tube into the ovule? A. Stigma, B. Style, C. Filiform apparatus, D. Embryo sac. Correct answer is C. Filiform apparatus. Explanation: the filiform apparatus, located in the synergid cells of the embryo sac, plays a crucial role in guiding the pollen tube toward the egg apparatus for fertilization. While the stigma and style are involved in receiving and supporting the growth of the pollen tube respectively, they do not provide directional guidance at the final stage. The embryo sac contains the egg but lacks the structural feature to direct the pollen tube. Thus, Only the Filiform apparatus actively guides the tube into the ovule. How can you test pollen-pistil compatibility in the lab? A. With sugar solution. B. By boiling the stigma. C. Using ethanol dip. D. Microscope alone. Correct answer is A. With sugar solution. Explanation. A common lab method to test pollen-pistil compatibility involves placing pollen grains on a sugar solution and observing pollen tube growth under a microscope. The sugar solution mimics the stigma's environment and promotes germination. Boiling the stigma or using ethanol would denature proteins and kill cells, making them unusable for compatibility testing. A microscope alone cannot facilitate germination. It can only be used to observe it once the correct conditions are provided. A plant breeder removes all anthers from a bisexual flower. What is the process called? A. Bagging. B. Cleistogamy. C. Emasculation. D. Crossbreeding. Correct answer is C. Emasculation. Explanation. The deliberate removal of anthers to prevent self-pollination is known as emasculation. This technique is fundamental in controlled breeding experiments. Bagging refers to the covering of the flower to prevent unwanted pollination, but does not involve removal of any parts. Cleistogamy is a natural condition where flowers self-pollinate without opening, which is unrelated. Crossbreeding is the broader process that might use emasculation, but does not describe this specific act. What is essential before doing pollination in artificial hybridization? A. Stigma maturity. B. Anther maturity. C. Ovary swelling. D. Sepal removal. Correct answer is A. Stigma maturity. Explanation. For successful pollination, the stigma must be mature and receptive, meaning it should be physiologically capable of recognizing and supporting the growth of pollen tubes. Pollinating a non-receptive stigma usually fails. While anther maturity is important for collecting viable pollen, it is not a prerequisite for receiving pollen. Ovary swelling typically occurs after fertilization, not before. Sepals have no direct role in pollination, and thus their removal is irrelevant. Why are emasculated flowers bagged immediately? A. To prevent dehydration. B. To trap heat. C. To avoid contamination from unwanted pollen. D. To promote nectar secretion. Correct answer is C. To avoid contamination from unwanted pollen. Explanation. Bagging immediately after emasculation is done to prevent accidental pollination by foreign or self-pollen, which could compromise the breeding program. Dehydration is not a significant issue at this stage. Trapping heat might negatively affect the flower's viability, and nectar secretion is unrelated to the purpose of bagging in artificial hybridization. To observe pollen tubes, one should use A. Sugar slide under microscope. B. Pollen grains in soil. C. Boiled pollen. D. Fertilized ovary. Correct answer is A. Sugar slide under microscope. Explanation. To observe pollen tube development, pollen grains are placed on a sugar solution slide, which mimics the stigma's surface and promotes germination. Under a microscope, the elongation of the tube can be visualized. Soil lacks the controlled conditions and transparency for such microscopic study. Boiled pollen is non-viable and incapable of germinating. A fertilized ovary represents a post-fertilization state and is unsuitable for observing pollen tube initiation.